So thanks everyone for being here and welcome to our FLUXNET-ECN webinar of the month. Today we're going to be hearing from Sam Murphy and Jason Horne on biogenic fluxes in urban environments. So I'll introduce our first speaker, Sam. Sam Murphy is a PhD student working with Dr. Kenneth Davis and Dr. Natasha Miles in the Department of Meteorology and Atmospheric Science at Penn State. Her research is part of the Indianapolis Flux Experiment, or INFLUX, and broadly, Sam's research interests include land-atmosphere interactions, urban greenhouse gas emissions, carbon cycling, and boundary layer meteorology. So, Sam, I'll let you take it from here.
Okay, great. I'll share my screen and get started. Okay, so hi everyone. As Helen said, I'm Sam, and today I'm going to be sharing a bit about my research project, which focuses on the question, can a simple ecosystem CO2 flux model help us understand complex urban background CO2 conditions for Indianapolis, Indiana? So to start off, I'm going to give a bit of an introduction to the Indianapolis Flux Experiment, or INFLUX. This project is a testbed for developing, evaluating, and improving the methods for measuring urban greenhouse gas emissions. So shown here is an overview of some of the ground-based measurement network. The black triangles represent the locations of mole fraction observations of CO2, and the red diamonds indicate locations of CO2 flux observations at agricultural sites. And so now this isn't the entirety of the influx network. There are other observations occurring here. For example, there are also urban and turfgrass CO2 fluxes, and you'll hear more about the turfgrass fluxes from my colleague Jason Horne when he presents next. So here is just a photo example of what one of the mole fraction tower observation sites look like. These are all on the communication towers. So here's just an example of one of them. And then here is a photo example of a CO2 flux observation site at a agricultural field. So one of the things that you can do with a network like INFLUX is to calculate the urban enhancements of greenhouse gases. So specifically today, I want to talk about CO2 enhancements. And there are a variety of different methods to do these calculations, but one way you can do it is to take the difference between two CO2 mole fraction observations, one of them being downwind of the city and one of them being upwind of the city. And this upwind observation is what we call the background. And we need to do this because we have to be able to separate the enhancements that are due to the greenhouse gas mole fractions that are emitted from within the city from the greenhouse gases that are coming into our domain. So while this is a pretty simple calculation, this recent study from our group by Miles et al. And co-authors found that the choice of background tower is not necessarily a simple one. So this study found that the choice of background tower significantly impacted the urban CO2 enhancement calculation. So in this study, they compared the CO2 enhancements calculated using 3 different greenhouse gas mole fraction observation towers in the INFLUX network. These towers are circled here over here on the right. These towers that they looked at were 1, 9, and 14. And in the background of this figure is the National Land Cover Database information for the year of 2019. So through this study, they found that there were growing season differences in CO2 mole fractions on the same order of magnitude as the urban enhancements themselves. And they assert that these differences are due to the impacts of the surrounding vegetation. So the authors were able to show that the differences between the Tower 1 and Tower 9 mole fractions observed could be explained by the expected differences in the timing and magnitude of the CO2 fluxes at deciduous broadleaf forest sites and agriculture sites, because Tower 1 is predominantly surrounded by forest and Tower 9 is predominantly surrounded by agriculture, which in this domain is mostly corn or soybean. However, they still notice these huge differences between Tower 9 and 14, which are both agriculture sites, or both predominantly surrounded by agriculture. So this led the authors to assert that we need to be able to quantify the seasonality and magnitude of biological CO2 fluxes in the region in order to interpret the tower-based observations. So this work led to my research question, which is how well does the vegetation, respiration, and photosynthesis model, or VPRM, capture these complex background CO2 conditions that have previously been observed in Indianapolis? So this research question has sort of two components. One of them is how well does the model capture the observed growing season CO2 fluxes, particularly at agricultural sites? And then how well does VPRM capture the observed growing season differences in CO2 mole fractions between background towers? So one component is looking at the CO2 fluxes, and then another component is looking at the CO2 mole fractions. So now I'll give you a bit of an overview of the methods that I use in this study. So quickly to summarize VPRM, there's a few inputs that are needed. You need the land surface water index and the enhanced vegetation index, which are both remotely sensed products. And then you also need land cover information, which for this study, we use the National Land Cover Database and cropland data layer. And then you also need photosynthetically active radiation and temperature, which for this study, we get both of those from WRF. So these things are fed into the model equations, which represent respiration and photosynthesis, along with model parameters developed using flux tower data for the different plant functional types in the model. So this leads to the output of respiration, gross ecosystem exchange, and net ecosystem exchange. For this study, we ran VPRM for a domain centered around Indianapolis, which is shown over here on the right. This domain is roughly 300 by 300 kilometers. We run it for a 1 kilometer squared spatial resolution and an hourly temporal resolution. And for this, we use the model structure and parameters developed by Gorgi et al. 2022. And we run this for a time period between 2017 through the end of 2021. Next, we convolved ecosystem CO2 fluxes from VPRM with tower influence functions, which represent the impact of the surface on the observations at the towers. We did this for 3 potential background towers in the influx network, towers 1, 9, and 14, the same exact towers that Miles et al. 2021 considered. And then this results in estimated components, biogenic components of the CO2 mole fraction for every hour. And we do this for the time period between April 2017 and March 2020. And that's just to account for the time periods when all the towers were operational, and also to exclude any time periods where the observations could be impacted by the COVID-19 lockdowns. Then after we do this, we calculated the observed enhancements and then the modeled enhancements for the background towers. So that the enhancements are defined like described here, which is the Tower 1 enhancement is defined as the difference between the Tower 1 CO2 and the Tower 9 CO2. And the Tower 14 enhancement is the difference between the Tower 14 CO2 and the Tower 9 CO2. And so I just want to note that these are not urban enhancements. These enhancements just refer to the differences between these towers. So then we compared the modeled fluxes to the observed fluxes. And to do this, we focused just on the growing season. So just June, July, August average daily cycles of CO2 flux. And to do this, we filtered the observed data using criteria from Vickers and Mart 1997 and Foken 2008. And then we also filtered to only include times when 90% or more of the flux footprint was attributable to the vegetation of interest. And so we did this using the flux footprint model from Kloon et al. 2015. And then down here is a summary of the sites that we used in this comparison. And I just want to point out that all the sites that we used were either corn or soybean sites. So then we also compared the CO2 enhancements, the model to the OBS. And to do this, we removed any times when the towers were impacted by the urban plume. And then we focused only on the afternoon average enhancements, so from 12 to 1700 local standard time. And then we wanted to assess VPRM's ability to capture the daily, monthly, and seasonal trends in enhancement observations. So now I'll show you some of the results from this study. So starting off with the model observation comparison for CO2 fluxes at agriculture sites. So here on the x-axis, this is the mean CO2 flux, so in micromoles per square meter per second. And then the x-axis is hour of the day. And then the model is in red and OBS are in black. And the top row shows all of the soybean sites considered, and these middle and bottom rows show the corn sites. And then the bars represent the standard error. So from this, we can see that the model captures the timing of the daily cycle of CO2, but its ability to capture the peak magnitude of drawdown during the afternoon varies, mostly for some of the corn sites like this one, I over here, or H. But its ability, sorry about that. Additionally, there are some times where it's not able to capture the respiration. So over here, on panel A, it doesn't fully capture the magnitude of the respiration in the observed. So moving on to the enhancement comparisons. So here in the top row, we have the modeled and observed afternoon average CO2 enhancements for every day in the time period we considered. So on the left here is Tower 1, and the right here is Tower 14. And then the associated residuals are shown in the bottom row. And the residuals are usually on the same order of magnitude as the observed enhancements themselves. So from this, we conclude that at a daily timescale, the model does not capture the observed average afternoon enhancements. So moving on to a monthly timescale, here we have the mean monthly residuals of the afternoon average enhancements for every month considered in our time series, with Tower 1 on the left and Tower 14 on the right. And the error bars represent 2 times the standard error. Most of the monthly residuals do not differ significantly from 0, so that's really exciting. So from this, we assert that there is no seasonal bias and that the model error is random. So at a monthly timescale, the model is much better able to capture our observations than at the daily timescale. So now considering how the model performs on a seasonal basis, we have a multi-year composite of 31-day running median afternoon average enhancements for Tower 1 in the top panel and Tower 14 in the bottom panel. So the model is able to capture the general timing of the growing season trends for both towers. However, I want to point out specifically for Tower 1, it doesn't capture the full magnitude of the enhancements early in the growing season. However, it does much better at capturing the magnitude of the enhancements for Tower 14. So moving away a little bit from the model observation comparisons, we also looked at the modeled afternoon average net ecosystem exchange within the influence function for each tower. This is just for 1 month for July 2018. And we did this for the 3 major vegetation types surrounding the towers. This is corn, other crops, and deciduous broadleaf forest. So the different towers are shown in the different colors here. And what I really want to point out is that there is a gradient in productivity predicted for the different towers, with Tower 14 having the largest predicted drawdown, so the most productivity, and Tower 9 having the least predicted drawdown, so the least productivity for all 3 of the PFTs considered here. So from this, we learned that the differences in the predicted mole fractions are a result of the differences in predicted productivity. So to wrap up my presentation, I want to summarize what we learned so far. So the model captures the trends in the daily cycle of the CO2 flux during the growing season, but it doesn't always capture the full magnitude. As we saw, sometimes it didn't capture the full magnitude of respiration, and for some sites, it didn't capture the full peak magnitude of the CO2 drawdown. And then thinking about the enhancements, the model wasn't really able to capture the observed CO2 enhancements at a daily time step, as given by the residuals being on the same order of magnitude as the observations themselves. However, at monthly and seasonal timescales, the model performed much better. And the model predicts differences in productivity across the different observation towers. So to sort of give you a so what and summarize and conclude everything in the presentation, we learned that VPRM can be used in urban CO2 inversions to represent complex seasonal variations in background conditions that have previously been observed in Indianapolis. And so these results are really encouraging for the use of VPRM in other urban settings. And so these are my references. And with that, I'll thank you for listening.
Great talk, Sam. We have some time for questions and discussion. So if anyone has any questions or comments, please go ahead. Well, if no one has a question yet, I have a question, Sam. You said you remove observations that are impacted by the urban plume. How did you go about doing that? Was that just covered by using the footprint model or a different way?
That was, we just filtered out by wind direction.
Okay, makes sense. Cool. Thanks.
Hello. I have one question.
Sure.
Yes, is there any parameter, say for example, seasonal wise, and they also change their carbon dioxide enhancement. So there are different parameters that are occurring during different seasons. Say for example, VOC, volatile organic compound, is there any role in carbon dioxide enhancement?
Sorry, I didn't quite catch the end of your question. Could you just repeat that?
Volatile organic compounds have some roles during in different types of the parameters. So in carbon dioxide enhancement, is there any role of volatile organic compounds for carbon dioxide enhancement?
We only used annual parameters for it.
But they have some, we can see that they have some roles in different seasons. So if carbon dioxide enhancement occurs during different seasons, so maybe they have some roles, but I do not know.
No, I'm sorry. I don't think we considered anything like that. I'm sorry. It's also just for some reason the audio is coming through a little quiet on my end, so it's a little hard for me to hear the question. I apologize for that.
Okay.
Oh, looks like we have a question in the chat from John Wang. Any ideas on why the drawdown magnitude was sometimes not captured well in the summer? How might the model be improved?
I see. I mean, from my understanding, sometimes even within one cornfield or one agriculture field, the productivity and drawdown can really vary, even just within that one field. So it might be hard to be able to capture the exact drawdown of every single site. But I guess one way that it can be improved could be just including, without like changing the actual model structure, would be to just include more sites in your parameterization.
Looks like another comment in the chat from Wu Sun. It appears the big respiration spikes in the soybean field are morning flush out of nighttime accumulated CO2, so probably not real. I don't know if it's a question or more just a comment.
Yeah, I'm not sure. I think it might just be a comment.
Hey, this is Wu. It's a comment about one of the figures you showed, which compares the observed flux versus modeled flux. I think for one of the soybean field, you had the respiration spikes just around daybreak, and I think that's because of the disintegration of nighttime residual layer, which flush out the accumulated CO2 at nighttime. So probably might not be real, I guess.
Okay, I see. That's possible, but I could take that into consideration and look into that a little bit more. I see another question in the chat.
So, can you hear me now?
Oh, hello.
Yes.
Yes. So, sorry, I just have a question. So, regarding the lower respiration of the VPM, so actually there's one modified version of VPM, and it seems in the paper it seems like it can better simulate the respiration, especially at nighttime. So, have you ever tried or have you looked into it? Because they also have parameter for, I think, also American, yeah.
Oh no, I haven't run another version. I've just used this recent version. Do you know what the version that takes into the account the higher respiration that you're saying?
There's a modified version of the VPM. The name of it is basically the modified VPM, and it changed the formula of the VPM respiration formula, because the standard VPM is just a linear correlation with the temperature, right?
That is
Oh,
Sharon.
this
That is Sharon's model.
yeah,
Yeah.
that is the one we're using. This one doesn't have the linear respiration.
Okay, so it still have a lower underestimated respiration, right? Okay, thank you very much.
Yeah. But I think it's much improved compared to the linear representation of respiration.
Thanks.
I see another question in the chat from David Miller. Any thoughts on how the higher temporal aspects of VPRM, e.g. daily, could be improved, other variables that could be considered?
Well, I guess one problem, I assume you're referring to sort of the comparison between the enhancements and how the daily really was not very good. But I guess part of the problem is it's not just VPRM, it's also the influence functions. So any sort of like discrepancy in the influence function, which I think can have errors in wind direction at time compared to observations, that could also lead to some of the discrepancies that we're seeing at the daily scale. So I think that if we wanted to improve this kind of quantification of this estimation of the enhancements, we'd have to also look at the influence functions and see what's going on with them outside of just modifying VPRM. Okay, great. Thank you for the question.
Okay, well, if no one else has any more questions, then I'll go ahead and introduce our second speaker. And then if you think of questions later, we can always come back to this discussion. So Jason is a PhD student, also in Penn State's Department of Meteorology and Atmospheric Science. He works with Dr. Ken Davis and Dr. Ying Pan, conducting research under Baltimore Social Environmental Collaborative, or BSEC-IFL, the Indianapolis Flux Experiment, and the Susquehanna Shale Hills Critical Zone Observatory. His current research focuses are meteorology, surface layer turbulence, microclimate modeling, biometeorology, and urban ecology. So go ahead, Jason.
Yeah, you've left me a nice chunk of time here too to chat. Thanks everybody for coming out and listening to this. It means a lot. This is kind of a really cool opportunity, so it's a pleasure. I see a few folks that I've talked to before and might have seen this talk in its infancy. Hopefully at this point it's more or less flushed out and this won't be a repeating of old information that you might have already seen. But some cool stuff. So it's kind of going to play off of what Sam just talked about, where we're using the VPRM to try and estimate biogenic fluxes in the urban area. But here we're seeing how well does this VPRM perform at replicating turfgrass fluxes at turfgrass flux towers that we have in Indianapolis? Because let's say that these turfgrass systems are not being well represented in the modeling framework, and you use this model for a prior in some sort of atmospheric inversion. Well, then it could cause additional errors in your estimated anthropogenic flux and things of that nature. But when Ken first came to me with this project and he said, Jason, I have this project looking at grass, turf grass specifically, I said, oh gosh. I said, turf grass, don't they have that saying where watching the grass grow, which doesn't necessarily coincide with something very fun and enjoyable. But again, like I just said, this has big implications in actually a larger framework of modeling and stuff like that. So let's get into it. Well, maybe if I can progress this slide, let's see. There we go. Okay. What'd you say? Huh?
Can't see the slides.
Oh, we can't see the slides. Good call. Nobody was going to stop me either. I was just going to go. Okay, can we see the slides now?
Yes.
Nice. Very good. Ella's sitting next to me if you didn't know that. Anyway, so here are some flux tower data, diel cycles of CO2 fluxes, kind of like what Sam had shown here. These are some of our flux towers out in Indianapolis. One big thing is, boom, we have an observed drawdown at our turfgrass sites in the middle of the winter. This is pulled out of Kai Wu's paper from 2022. And now, like I was saying, let's say that this turfgrass photosynthetic drawdown is not represented in the modeling framework. Well, this could cause problems. And it just so happens that the VPRM model cuts off photosynthetic activity if you go below 0 degrees Celsius. Well, I'll tell you right now, during these winter periods, it got below 0 degrees Celsius quite a bit. So this is kind of a problem red flag here. It was like, well, we need to implement and maybe change this parameterization now so we can allow photosynthesis at a lower than 0 degrees Celsius. So let's get into it a little more. Same thing that Sam showed here. This is NLCD, National Land Cover Database, 30-meter resolution, land surface types. All the red is the urban land cover. And essentially what the VPRM does is it assigns, wow, it is really not progressing when I want it to progress. Let's see, there it goes. Essentially what it does is it assigns some sort of PFT with a set of parameters for the specific equation to each of these cells. And in the original model framework, and again, this is from Sharon's model from 2022, you can see this is Monroe Morgan State Forest, deciduous broadleaf forest, and then outside here, we've got corn parameters. But inside, this is where we're specific, inside the city, in the original modeling framework, open developed spaces are represented with grass pasture parameters. And then anything in the rest of the innards of the urban areas, this is low, medium, and high developed for those who are familiar with the NLCD data. These are represented with deciduous broadleaf forests. And well, here's my question. What happens if I take and I replace these grass pasture parameters for these open urban areas and I use a turf grass parameterization that I make? And then let's say it's winter when the trees have no foliage, and underneath is actually exposed turfgrass lawns. And even in these innards of the urban areas, here in the centers and stuff like that. What if we represent that period with turfgrass parameters too? What does that mean for our quantification of the total biogenic urban flux? How does that change things? So first question we should ask is, what even is turfgrass really? I keep saying this, but what makes this any different than a regular grass ecosystem? Well, it's not really much different as far as species composition than a lot of times from a regular grass system. Of course, you can always have introduced species, like at a golf course or something of that nature. But really what makes this different from your regular natural, in quotes, grass community is the aspect of urbanization and artificial management from humans. So again, this player of urbanization, artificial management is probably what is going to distinguish this from a normal grass pasture parameter. But the question hasn't even really been answered yet. Can we just model turf grass as regular grass pastures in the first place? So here's our two turfgrass flux towers that we had in Indianapolis. USINA, these are their Ameriflux FLUXNet names. USINA was out at a cemetery, and USINB was in a golf course. And so you can kind of see with these two turfgrass towers, we actually capture a spectrum of management in the urban area too, you can kind of think. Whereas at our golf course, you would assume a higher, more intense management regime than you would at your cemetery. So using these two towers, we're also kind of looking at, well, can we kind of pick out any impacts of management, particularly at these turfgrass towers? And we'll kind of see this a little bit as we get into it a bit here in a moment. Let's move forward. But really, like, who cares about the turfgrass in the first place? Like, how much turfgrass really is there in Indianapolis? Well, I use this simple linear relationship with impervious surface cover that I also took from the National Land Cover Database, 30-meter resolution. This was developed by Melessi et al. Back in an early paper in 2005. And I used that to estimate turfgrass at each of the 30-meter pixels. And you can see from my calculations, roughly 33% of Indianapolis, the urban area I'm talking about. So if the pixel was categorized as open developed, low, medium, or high intensity, 33% of that area is covered in turfgrass. This is obviously a first order approximation estimate. We could do better. Actually, David's in here and he did a really cool study where he used LiDAR data too to kind of break out trees from turfgrass stuff. But here we really want to try and just look at a first order approximation of the carbon impact of the turfgrass. And I didn't actually believe that this would do a really good job at first until I started looking and peering into it a little further. And it does a really, really nice job for just a simple linear relationship. So anyway, so now we have an idea of how much turfgrass is out there in Indianapolis in this domain. So this is also our domain, I should specify, because eventually what we're leading up to is we're going to run a whole domain level analysis of this like city, Indianapolis. Let's move forward. Boom. So I'm going to brush over the optimization stuff. If anyone's curious, we can talk about it a little bit after the presentation, but I used a differential evolution algorithm to optimize the model parameters to our observed turfgrass data, following a lot of the same methodologies that Sharon did. And I also did this because I wanted to kind of cover my tracks a little bit. So how we're going to do this model analysis is I'm first going to test out the deciduous broadleaf forest parameters and the grass pasture parameters that Sharon developed in her model and see if they can replicate the observed photosynthetic drawdown and overall the entire diel cycle at our turfgrass sites. But then I said, well, what happens if I optimize those same observed data that Sharon used to make the deciduous broadleaf forest and the grass pasture parameters, but I used my optimization strategy? Does that change anything? And by that, I let them photosynthesize below 0 degrees Celsius. Can they then capture it, and does it actually make a difference? Do we need a turfgrass parameters? Do we need this turfgrass PFT? And then, of course, I actually make turfgrass PFTs, which each of our turfgrass sites, where I actually take the annual whole time series and I optimize a set of parameters with that. And then I also, as you'll see here in a second, split the time series into 2 seasons, and I make a dynamic set of parameters that changes with season to try and replicate the turfgrass towers. All right, enough rambling, let's get into it. One thing I should also hit on before we move forward though, is this is both of our turfgrass towers and their fluxes, just an average diel flux for an overlapping period when they were both out there. And what's interesting is you don't see a particular difference in these two. This net drawdown we're noticing here, this photosynthetic drawdown, it's not looking very different. So this was surprising, because I thought for sure you have this managed system, this golf course, you would have observed some sort of larger drawdown. Now, mind you, this also is during the middle of winter, and it only gets into that early part of April. So it's unclear as to when they were doing it and implementing these management things, but it might suggest that it is a short-lived life for potential management strategies on net ecosystem exchange. And then once you actually get outside the realm of when you apply these managements, that it could be not impactful on the photosynthetic drawdown. That's just a hypothesis. It really was very interesting, though, that we couldn't see any difference here in the middle of the winter between these two. But as I'll show here in a moment, this does cause problems for the modeling framework, for sure. First step, let's see how well the deciduous broadleaf forest parameters and the grass pasture parameters perform. Blue is our observed data. These are the various parameter sets, deciduous broadleaf forest, grass pastures, and then these are the two that I re-optimized with my models, my optimization methods. And you can see, oh, wow. Neither of them can really catch that magnitude drawdown that we were observing at that turf grass tower. So this kind of suggests to me that maybe we do need some sort of specialized turf grass parameter set. So now here's that same curve, observed data, but I'm now using the cemetery parameter set and the cemetery seasonal parameter set and the golf course parameter set. And again, the cemetery season versus cemetery is one I used the entire time series, not even regarding any sort of seasonality, and I optimized a set of parameters with that. While the other one, I split into an arbitrary cool season and warm season. So you have this dynamic set of parameters that switches when you're in the cool season and warm seasons, which kind of has a rough following of grass phenology. But you can see, obviously, one thing here is that we require some sort of seasonal parameter set to model these cool season fluxes. And I should also say this is only during the cool season, which is that end of November to beginning of April period that I showed before. So this is only the cool season in quotes. But you see, I need this seasonal parameter set in order to catch the magnitude of that drawdown during this period. One other thing to specify is that if I take this seasonal parameter set and then I put it over in my golf course, it doubles, it is essentially doubles the magnitude drawdown that it models at that site. And what this is because those two flux magnitudes are roughly the same as I showed before, and you suddenly put this parameter set in the golf course EVI is much higher. So the golf course is much greener than the cemetery. Suddenly you take this parameter sets that were limited by greenness and you put them in a green situation, they overestimate it. And then the exact opposite happens over here on the other side. Whereas if I take my golf course parameter set, which was optimized in this very green scenario with roughly the same flux magnitude, put it over here, it underestimates it. So this could be suggesting some amount of management impact, and it's at least adding confounding results to the VPRM. So now I'm going to start looking just really quick at the warm season. And the golf course was only deployed during the cool season, so I'm not able to use that here particularly and compare that to observed golf data. But I have cemetery here. First off, how well do the deciduous broadleaf forest parameters and the grass pasture parameters fare? Well, one thing to note right at first is it seems that with my grass pasture parameters, I am able to catch that drawdown in the middle of summer. So this might suggest at first glance, well, maybe grass is just grass in the middle of summer and something is differing in these cool season months. But if you take a look just for another and glance over here, all of a sudden you realize it is not capturing that nighttime respiration magnitudes that we're observing at the tower. And this is a big problem. So if I was to integrate across this entire diel cycle with my grass pasture parameters, it models the turf grass lawn as a net sink, when in reality, if I integrate across the observed data, it is a net source to the atmosphere. So if you're trying to represent carbon dynamics with these biological communities in an urban area, that could be a big problem. First thing here, now we're using the turf grass parameters, and you see the golf course parameters do fairly poor because obviously they're being used outside the realm which they were optimized. But here are seasonal parameters. They both perform fairly well at capturing this diel cycle. So moving forward, given that they perform the same here in the summer and that I need the seasonal parameters in the winter, I'm going to continue to use these symmetry seasonal parameters when modeling turf grass across the entire domain, with the assumption as well that there's more symmetry lawns or lawns like symmetry than there are golf course lawns. Now, I could take this a step further potentially and isolate each of the golf courses and represent them with golf course parameter set. But for this study, we just want to look at what is a first order estimate of this carbon impact of these turf grasses in Indianapolis, in these urban areas. So some of the takeaways from this site level summary is that deciduous broadleaf forest parameters and grass pasture parameters are not able to calculate or capture these turf grass cycles of CO2, at least the diel cycles. And particularly the cool season, I need a seasonal set of parameters in order to model this turf grass CO2 flux. And again, the golf course and the cemeteries require a different set of parameters in order to capture those cool season CO2 fluxes. So now let's move into a little bit of the, big takeaway. Turf grass should be uniquely represented in the VPRM modeling framework. So let's move across the entire domain. Same domain I showed you guys before with the turf grass map. What am I at time? Oh, I'm pretty good on time. That's nice. So I have this picture over here of the domain because I want to explain this figure. I'm talking about urban vegetation. And by urban vegetation, I mean all the green. So those are the urban vegetation. Whereas obviously there's vegetation outside of here, but I categorize that as non-urban vegetation. I specifically want to look at the urban vegetation here. So average daily net ecosystem exchange across this entire domain for the urban vegetation, you can see I'm comparing VPRM model with no turf grass and a VPRM model with the turf grass represented. In the cool season, you can see the actual net magnitude source goes down by half if turf grass is represented properly. And obviously that's because photosynthetic activity is now being represented. So what this could mean potentially is that if you are going and you're using this period for a biological prior, you are overestimating the biological contribution, then maybe potentially underestimating if you're trying to solve for the anthropogenic emissions. Another thing to point out, pretty blatant and obvious right here, is that if I don't represent the turf grass properly in the warm season, it's actually just completely changing the source sign from a net sink to a net source. Kind of like how I was talking about the grass pasture versus the turf grass parameters in the site level description. But this is very interesting, but it also kind of reinforces that, yes, we probably do need to represent these turf grass communities uniquely in this specific modeling framework, but it also might carry over into other modeling, land surface models like NOAA-MP. This is kind of the direction we're going to be heading potentially with the BSEC project, is we're going to see, well, we kind of show that maybe these things do need uniquely represented in modeling frameworks. Where else can we make improvements to the modeling structure or the parameterization, et cetera, et cetera? So what does this mean for other studies that have kind of used this VPRM for either like a biological prior or made some assumption about the biology? I'm going to look at comparatively to a study done by Turnbull and others in 2019, where they compared multiple methodologies for quantifying whole domain CO2 flux, and the domain just so happened to be, well, obviously I made it this way, the exact same domain as I ran my VPRM in. And in Jocelyn's comparison, she assumes that this biological flux is roughly around 3,000 moles per second, which if you didn't know any photosynthetic activity is going on, it's not a bad assumption. But suddenly when we started measuring turf grass fluxes, we said, uh-oh, this might be a problem. And comparatively, the estimated whole city fossil fuel emissions is 19,000 moles per second. So let's see here. Turnbull et al., the biogenic source estimated for my non-urban vegetation is roughly the same as the value that she estimated. However, in the urban areas, we can see this urban turf grass is acting as a net sink. So I should have explained this a little more too. This is for a very small window of time here for the 17th to the third when Jocelyn could make these comparisons between multiple methods. And it's also only during these afternoon hours, and that has to deal with boundary layer mixing, convection, and stuff like that. We're not going to get into that so much, but that's specifically why I chose this period and these times. But you can see that the blue is both of these two, the orange being the urban and the green being the non-urban. The blue is the both of them combined, and the shading here is the range of values that it took on in the modeling framework for each of those hours. So this is max min for hour 11 in this period. And you can see a lot of times in the afternoon, the whole domain is a net sink for the biology. And again, if you are not accurately representing this turfgrass community, and assuming it's just this constant biological flux here, then you could be introducing additional complications and maybe errors in sort of methodologies that use this and need this information to estimate anthropogenic and or total CO2, total urban CO2. So, all right, that's all I had. I'm going to open it up for questions. I know that was probably a lot and going all over the place. Again, I really appreciate you guys for coming out here and listening for this whole thing. And at this point, I will take any questions because I'm sure there are a few out there. Thank you, guys.
Yeah, nice talk, Jason. Looks like we still have about 15 minutes, so if people have questions, please speak up.
Oh my goodness. Oh my gosh, so much in the chat.
We were chattering, sorry.
Yeah, I see this now. I don't know how you want to do this, Helen. You run this how you want to.
Well, it looks like we have a question from Teresia, so go ahead.
Hello, Jason. Thank you for the talk. I just have one question about incorporating your work in models. So you mentioned land surface models. So your work would be included in the urban modeling part or the vegetation modeling part, like how, I mean, I don't know if you have thought about that. I mean,
Sure. And I don't know. I guess I should have made the claim that I might not necessarily be doing this, but yes, the work would kind of be going to guiding land surface models, something like NOAA-MP or ELM, a large climate model, and kind of looking at how we can potentially, or what things that might need to be represented in this modeling framework to continue to represent these urban biological communities accurately, if that makes any sense.
Yeah.
Okay, cool.
Okay, so we're getting a few questions in the chat. First one from John Wang. Maybe I missed it, but isn't turfgrass covered by snow in the winter, and doesn't that impact the remote sensing?
Well, sure. It depends on where you are as a latitudinal. I mean, here in Indianapolis, there's large periods in the winter when there's grass is not at all covered in snow. However, if you go up to Minnesota, for instance, where David was doing his work, and you look at the Kumo Tower, I mean, that thing's almost covered in snow the entire winter. Correct me if I'm wrong, David, but so you don't really see any sort of net drawdown during that period. But here in the Indianapolis towers, there's a large periods when it's not covered. But the answer to the question about remote sensing and snow complications, yes, that definitely introduces complications for the indices that the VPRM uses when you use, or when snow is present on the surface. I don't know if that answers the question at all. Oh my gosh, it's blowing up. I can't keep up with this.
Okay, so here's another question in the chat. From what sources are the vegetation indices, meaning EVI and LSWI, calculated? Are they derived from MODIS at 500 meter resolution or from higher resolution Sentinel data?
This is a good question.
Oh, there's more.
Oh, good. Go for it. Sorry.
Could the potential representation errors in these vegetation indices have an impact on VPRM modeled fluxes when compared with locally observed in situ fluxes?
Absolutely. The question is for that last part is absolutely. The answer is 250-meter resolution MODIS data. So one of the questions that we're kind of pushing forward here and maybe asking, I've thought about maybe implementing it in Baltimore, is using high resolution Sentinel 10-meter data and or a hybrid Sentinel Planet product that could potentially resolve those small scale heterogeneities that exist in the urban environment. But you would think, yes, that might be impacting this somewhat, where you have sort of an EVI dilution if you've got this mosaic within that one cell. I think that answers your question, but I'm not sure. Continue if it did not. Oh yeah, David put his paper in there. That's a good paper. If anyone's interested in turfgrass stuff, I would definitely read that one as well. I'm hopefully going to have this manuscript all put together here in the next few months. So look for that one to come up out as well. That's true. What Ken just put in the comments, I didn't talk about this, but going back to the pixel question and choosing a representative pixel, I did a lot of work specifically looking at the pixels overlapping that area and which pixel encompassed the most of my site at that particular moment, and which probably had the most representation and littlest amount of dilution from impervious surface, for instance. So I did a lot of poking around on that. Not perfect by any means, and it could only really be remedied by using higher spatial resolution data. But there was an effort put forward to try and get the most representative pixel for that specific site. I don't know. I might have missed another question here somewhere.
There was a lot of discussion in the chat earlier, but it seemed like a lot of people answered each other's questions. So I didn't read those. But if anyone has more questions, please go ahead.
We were chatting a lot, Jason, about why is the golf course so darn green? What's going on there?
Oh, I know. It's a good question though, too. I think it's because both sites in the winter are light and not temperature limited, or light and temperature limited and not nutrient and water limited. That's not a bad hypothesis too. But yeah, that's a question that's kind of made me scratch my head a little bit, and it makes me think, I want some more golf course data. And one more thing, obviously, I didn't get to include this in here, but I did a multi-site comparison between, we have another turfgrass tower that is not included in this that we have out at the NIST campus, outside of DC. And also I took the Kumo tower and I did seasonal comparisons and a whole bunch of other things. So, and that showed some interesting things as well, because I hypothesized that we are potentially able to see the impact of invasive species in our NIST turfgrass lawn, because there's a huge community of Japanese stiltgrass that is taking hold in that lawn. Whereas if you compare it to, quote, less impacted lawns like our cemetery, the fluxes look completely different. So it's interesting, but obviously I don't have enough time to cover all that stuff in this short period.
Go ahead, Elliot.
Hey, Jason.
Yeah.
Just a question out of curiosity. I don't really know much about some of the inner workings of the VPRM models, but especially like with the sites, kind of measuring the fluxes on the golf course where it's a fairly open area versus, say, some of the urban areas like towards the city center that may have compact houses. Does the model essentially resolve, especially like during winter when the sun angle is low and there may be some kind of shading and indirect light in a lot of those places? Would that make a difference?
So you're talking about like building shading, impacting
Yeah, some of
remote
the
indices, things like that.
basically to the amount of photosynthesis like at some of the turf grass, basically more so in like residential neighborhoods where
I
you
think I
have
see what you're
buildings
saying.
instead of like large open areas such as the golf course.
Yeah, and the answer to your question, yes. If there's
Mhm.
ever some sort of shading effect during particular sun angles, then
Mhm.
that would impact the photosynthesis, the
Mhm.
photosynthetic. But that is not like, this is a very simple model,
Mhm.
like at the
Gotcha.
core, and that is not being like accounted for here.
Gotcha. And no worries, just curious about it.
Yeah, yeah, absolutely. No, good question. Yeah, Ken, just before we go, unless people need to jump off, the Japanese stiltgrass is really interesting because it's a C4 warm season grass, so it acts as like evil corn. As soon as it pops up, because you've got this annual grass, it's like live free and die fast, and just starts sucking CO2 out of the atmosphere, giving you these crazy drawdowns in the middle of summer when you would expect the cool season grasses to kind of like get a little suppressed as far as their photosynthetic activity. So that's really interesting too. But you can see it's this crazy drawdown right now. I sent a photo to our group earlier because it's pulling out like 35 something micromoles per meter squared per second right now over the past few days in the middle of August, this turfgrass lawn.
Well, if no one has more questions, thank you everyone for coming, and thanks Jason and Sam for your talks, and thanks to FLUXNET-ECN and Christine for holding the webinar. And
Yeah,
Thanks
thank you
for
guys all
listening,
again for coming out and
everyone.
listening. Yeah, I appreciate that.